Okay, well, thank you very much for the invitation, and very sorry not to be seeing everyone in person. So I'm going to be talking about ATP receptors, lysosome function, and breast cancer. And in particular, I'm going to be talking about one receptor, which is P2x4, which is a member of the P2X receptor family. And so it's a cation channel activated by extracellular ATP, and it has very high calcium permeability in addition to sodium and potassium permeability. So P2x4 is the most widely distributed of all the P2X receptors. So it's been mostly studied in immune cells, such as microglia and macrophage, but it's also in the nervous system, it's in the heart, lungs, kidney, endothelial cells, epithelial cells, and it's upregulated in disease. So disease associated with inflammation, you get an upregulation of P2x4. And as a consequence, there's been a lot of interest over the years from pharmaceutical companies in developing ligands that target P2x4, mostly for treatment of chronic pain, both neuropathic and inflammatory pain. But there's also interest in its possible cardioprotective role in treatment of heart disease. There's interest in its use as a drug target for treating alcohol use disorders, and also very recently for its potential benefits in promoting remyelination in multiple sclerosis. Now, so there's interest in both positive and negative allosteric modulators of P2x4, but in general, the drug discovery efforts have been challenging, and progress has been a lot slower than it has been, for example, at P2x3 and P2x7. So I'm going to end my talk by very briefly saying something about our drug discovery efforts. But for the majority of my talk, I want to talk about the role of P2x4 in lysosomal function. So we've been very interested in understanding how P2x4 controls lysosomal membrane traffic, particularly autophagy and lysosome exocytosis. And this stems from observations years ago that P2x4 has this unique subcellular distribution that's not shared by other members of the family, which is that it's predominantly expressed in endolysosomes. And in most cells, expression at the cell surface is very low. So in some cells, such as microglia, P2x4 is trafficking very rapidly between the cell surface and lysosomal compartments. And in other cells, such as macrophage, it seems to be very much stuck in the lysosome. So this suggested to us that P2x4 has this dual role, both in sensing extracellular ATP at the cell surface, but also in controlling lysosome function. So this slide just shows some of the early pictures that images that we obtained expressing P2x4 here in just a single normal rat kidney cell. And what you can see is the P2x4 is in these enlarged vacuolar lysosomes, and they're enlarged because the cell is overexpressing P2x4, and P2x4 promotes lysosome fusion. And in addition, when we looked at the native receptor in different cells, here we've got mouse macrophage, again, we found the P2x4 inside in compartments that co-label with a lysosomal marker, LAMP1, and which label rather poorly with markers for early endosomes. So this was the evidence we had that P2x4 was predominantly in the endolysosomal system. And at that time, there was relatively little known about lysosomal ion channels. But in the last sort of 10, 15 years, this field has really expanded. And many different ion channels have been identified at the lysosomal membrane, several of which are permeable to calcium and sodium. So we have the TRPLs and the TPC channels in addition to P2x4. There's also voltage-gated calcium channels. and then there's a couple of potassium channels down here. So the lysosomes are acidic calcium stores. So inside the lysosome, calcium is quite high. It's of the order of 0.5 millimolar. So when the calcium permeable channels open, you get an efflux of calcium from the lysosome, and that calcium signal in the immediate vicinity of the lysosome is what triggers the lysosomal membrane traffic. It also regulates gene transcription, particularly genes involved in autophagy. So also within the lysosome, there's high luminal ATP, so that's relevant for the activation of P2X4, because the orientation of P2X4 is that its ATP binding site is luminal. And ATP is actively transported, driven by the membrane potential across the lysosomal membrane. And of course, the lysosomes are typically very acidic, driven by the proton ATPase. So we were interested, having identified the P2X4 within the lysosomes, where it's extremely stable, we were interested in understanding its role within the lysosomes and how it's regulated. And so we collaborated with a colleague at Dalhousie University, Jianping Dong, and he had developed methods for patch clamping lysosomes. And from those patch clamp experiments, we were able to show that we could record a P2X4 mediated current that was sensitive to ATP and also sensitive to pH. So the pH sensitivity of P2X4 had been known for many years. So it's strongly inhibited by acidic pH. So within the lysosome, the trigger for activating P2X4 is probably a shift in pH, alkalinization of the pH, rather than a change in ATP levels. So we're interested in what might trigger alkalinization of the lysosome and hence recruit P2X4 receptor activity. One of the stimuli that we've been looking at is the activation of the P2X7 receptor by ATP. And we have evidence that when you activate P2X7, you trigger alkalinization of the lysosome, and that recruits those lysosomal P2X4 receptors. But also, lysosomes are a heterogeneous compartment. So although we think of them as acidic, what others have shown is that the perinuclear lysosomes are very acidic, but that as lysosomes traffic towards the cell surface, they become much more alkalinized. And if you look at the images on the right-hand side of the slide, what we show here, so we took images of lysosomes using the confocal microscope, where we're taking a slice through the middle of the cell. So we're looking at perinuclear lysosomes. And then we took images using turf microscopy, where we're just looking at those lysosomes right in the vicinity of the plasma membrane. And we've labeled them with P2X4, green, and we've also labeled them with the reagent magic red. Now, magic red fluoresces dependent upon the activity of the lysosomal enzyme cathepsin. And that activity is dependent upon pH. So the amount of red fluorescence is an indicator of the pH. So what you can see here is that those perinuclear lysosomes that we're imaging with the confocal are quite red, but those at the surface are much less acidic. So what this suggests is that as lysosomes travel from the perinuclear region towards the cell surface, they become more alkalinized, they can recruit P2X4 receptor activity, and P2X4 receptor can then be involved in promoting lysosome fusion. So that calcium efflux mediated by P2X4 will trigger lysosome fusion. So lysosome fusion mediated by P2X4 is dependent upon calmodulin. So what we're looking here, the images on the left, again, these are images of lysosomes labeled with P2X4. And if you pharmacologically alkalinize the lysosomes with methylamine, then you can activate P2X4. And you see on the bottom panel on the left that the lysosomes are a lot bigger. So that's because they've undergone fusion. And that process is regulated by expression of calmodulin, middle panel, or by expression of a mutant calmodulin, right-hand side panel. So calmodulin interacts with P2x4 in a calcium-dependent manner to trigger lysosome fusion. And interestingly, what Jianping Dong has shown in a more recent study is that another lysosomal calcium channel, TRPML1, which also interacts with calmodulin, but it mediates fission. So you have this sort of antagonistic control of lysosome membrane traffic by these two calcium channels. So we have been interested in understanding the cellular context by which P2x4 mediated lysosome fusion, under what are the contexts where it plays an important role, and how does this contribute to its role in disease? So one context where P2x4 mediated lysosome fusion is known to play an important role has been identified from nice work by Manfred Frick's group at University of Ulm. And they have looked at P2x4 receptor in the lung, in particular in these type 2 pneumocytes. And these are the cells that secrete surfactant, and surfactant is very important for lung function. And the surfactant, if you look on the right, the diagram on the right-hand side, is stored in secretory lysosomes. And what he showed is that the secretion of the surfactant, so the exocytosis, is dependent on a P2x4 mediated calcium signal. So the P2x4 is localized to those secretory lysosomes, and its activation is required for that secretion. And this is potentially interesting when we think about COVID-19, because COVID-19 is associated with loss of surfactant function. That's contributed to this acute lung injury that's seen in patients. And there's recent data suggesting that ivermectin possibly has a role in treatment of COVID-19, although the data is by no means conclusive. And ivermectin is a well-known positive allosteric modulator of P2x4. So we would expect it to promote the secretion of surfactant. I mean, ivermectin obviously has many other targets. Another role for lysosomal P2x4 that's recently been reported is a paper from Matute and Domecq's groups in Spain. And they reported that P2x4 in microglia, when you enhance its function, contributes to the enhanced phagocytosis of the microglia, and that helps clear the myelin debris and promotes remyelination. And they did some pharmacology to show that promoting activity was beneficial, inhibiting activity exacerbated the disease. So we have two contexts where lysosomal P2x4 receptors seem to be involved. What we have focused on, though, is a role for lysosomal P2x4 in cancer cells. And the reason we were particularly interested in a role in cancer is because cancer cells have altered lysosomal function, and that altered function appears to be very important for promoting tumor progression and metastases. So lysosomes in cancer cells have more lysosomal enzyme activity, so cathepsin activity is increased. They fuse more, so they're more secretory. So you get release of the cathepsins into the extracellular space, which is thought to promote degradation of the extracellular matrix and also angiogenesis. And that's thought to be important for enabling the cancer cells to escape the primary tumor and to invade. And also because you have enhanced enzyme activity and enhanced lysosome membrane traffic, this contributes to enhanced autophagic flux. So autophagy is where the cell degrades itself, damaged organelles and macromolecules. And this is very important for contributing energy to the cell, particularly for a cancer cell, which is often where you have the cancer growing often in conditions of nutrient deprivation. So as a collaboration with Sébastien Roger at the University of Tours in France, we've been looking at P2X4 expression in breast cancer and the role that P2X4 is playing here. And on the left-hand side, what is plotted is just a demonstration that from a gene expression analysis, we see that P2X4 at the transcript level is upregulated, whereas several other of the P2X receptors are downregulated in cancer. And we also saw an increase in P2X4 protein in human breast cancer samples versus normal breast tissue detected by immunohistochemistry. So breast cancer and other cancers as well is associated with an upregulation of P2X4 receptor expression. So we wanted to see how changes in P2X4 receptor expression affect the ability of the tumour to grow and form metastases. So we used a fairly commonly used model, a mouse model of breast cancer, where we use these 41 cells, which is highly invasive mouse mammary tumour cells. They express P2X4. So on the left, you see this immunostain for P2X4, which shows, and we show that this is in the lysosomes. And these cells are implanted into mammary fat pad of a mouse, and then we wait 6, 7, 8 weeks to look at tumour growth and formation of metastases. And so we did this with our control cells and then with cells where we had knocked down P2X4 receptor with the CRISPR-Cas9 system. And essentially what we observed, if you look at the pictures on the top left, is that when we implanted the control cells over a series of weeks, the tumour grew. But when we implanted 2 different clones where we'd knocked down P2X4 receptor expression, the tumour growth was substantially suppressed. And also when we looked at metastases formation, so that's bottom left, there was almost undetectable metastases with our cells where we've knocked down P2X4 compared to the control cells. So it looks then that we have an upregulation of P2X4 in cancer, and then if you suppress that, you have quite a profound effect on the ability of the tumour to grow and metastasize. So we wanted to see whether this effect of P2X4 receptor expression in the 41 cells, whether it altered the lysosomal properties of these cells. So we compared our wild-type 41 cells with the cells where P2X4 had been knocked down. And our prediction was that by knocking down P2X4, we would expect to inhibit lysosome fusion. So that's shown in the diagram on the left. So if fusion is inhibited, but fission carries on, our prediction is that we would expect that knockdown of P2X4 causes an increase in the number of lysosomes. We'd also expect it to reduce autophagic flux because we're inhibiting lysosome fusion with the autophagosome. We'd expect to see a reduction in the secretion of lysosomal content because we're inhibiting lysosome fusion with the plasma membrane. And by inhibiting that secretion, we expect to see reduced cell invasiveness. And I've just got time to show you some of the data that we have on this. So on the graph on the left, we're plotting number of LAMP1 positive vesicles per cell as a measure of lysosome number. And indeed, when we knock down P2X4 in the blue, you see this increase in the number of lysosomes. We measured autophagic flux by measuring P62 and LC32, and we saw that in both our clones where we have P2X4 knocked down, there was an inhibition of autophagic flux. We looked at secretion of the lysosomal cathepsins. These are the degrading enzymes within the lysosome. On the left-hand side, you can see a Western blot looking at extracellular and intracellular levels of cathepsin D. And then the graph next to that shows that with the cells where we've knocked down P2x4, there's a reduction in extracellular levels of cathepsin D. And so we would predict from this that if that we'd expect to see less cell invasiveness, because the idea is that the cathepsin degrades the extracellular matrix and helps enable the cells to escape the primary tumor. So we did a simple 2D in vitro assay where we just looked at cell invasiveness through Matrigel, you see in the diagram top right, and just measured the cell accumulation in the bottom chamber. And we see that knocking down P2x4, you get a clear inhibition of cell invasiveness. And we also showed that we could rescue this by overexpression of P2x4 back into these cells. And we also got similar results with the P2x4 receptor antagonist BDBD, 5-BDBD. So just to then conclude our findings, so this is a paper that we just recently submitted, we feel that we have evidence that the upregulation of P2x4 that's associated with cancer alters lysosomal properties in such a way that it promotes autophagy, it promotes lysosomal secretion, and thereby promotes tumor growth and metastases. So I just want to end by saying something about drug discovery targeting P2x4. So hopefully from what I've said today, you can see that there's interest in developing both positive and negative allosteric modulators of the P2x4 receptor. So we carried out a study, so this was led by Andy Thompson, to identify novel modulators of P2x4 and P2x1, and this involved a screen of a fragment library. And from this, we've identified several new modulators with very low affinity, but we intend to take this forward as a collaboration with Leanne Stokes, who's at University of East Anglia, who's also been very interested in identifying new modulators of P2x4. But one of the interesting aspects of this study was that in addition to doing a screen of a fragment library, we also tested some of the recently published new modulators of P2x4. And we tested them by electrophysiology. And several of these, those indicated with the red squares around them, they've been published without, based on fluorescence assays, without any electrophysiological data. And when we tested them with our assays, we didn't see any activity. So whether that's, suggests that they require a longer incubation to have activity, we don't know. But I think it does highlight the need to carry out electrophysiological assays when identifying novel modulators of ion channels. So I just want to end by acknowledging those who've contributed to this work. So I mentioned that the work on breast cancer was a collaboration with Sebastian Roger, and all the work was carried out by Stephanie Sade, who was a postdoc in my lab for 2 years and has now moved back to University of Tours. There's a couple of students, former PhD students, Omar Qureshi, who now works in Solentech, Sin Lee Tan, and they contributed some of the work that I mentioned at the beginning of my talk. Jianping Dong, I mentioned, did the patch clamp of lysosomes and is a long-term collaborator. And then Andy Thompson, who worked with me when I was in Cambridge before I moved to Sussex, and now has his new company that he's started up, SI Drug Discovery. Okay, thank you very much.
Thanks, Ruth. That was lovely. Really nice data, and you know, obviously complex recording capabilities on lysosomes and on you know, obviously all towards cancer. I mean, on the lysosomal recordings, I don't want to sort of go into this in too much detail because we don't have the time, but we have actually been doing some organella recordings with AXAM. So Jean-Francois Roland, who I believe is on the call as well, or in the meeting, he's been doing some work with us to try and do automated patch clamp recordings of lysosomes and mitochondria. So watch this space as well for that. But anyway, we should move on because you've got plenty of questions, unsurprisingly, Ruth. So Mustafa Jamgoz asks, do you think the anti-metastatic effect could be coming from suppression of primary tumorigenesis?
Yes.
Okay, that's a really simple question. I like those questions.
I think that we're not really in a position to comment that there was such an inhibition of tumour growth that this could definitely be also suppressing metastases. But you know, just with the in vitro experiments, we've shown that there's several changes to the 41 cells, which would be consistent with them being less invasive. And that's what we see in vitro. So it's at least that data is consistent.
Okay, and Savi Gentili asks, as autophagy is important for recycling damaged mitochondria, do you see an increased ROS production in P2x4 knockouts?
I don't know the answer to that. That's a good question. Yeah. I don't know if anyone's looked, actually. I mean, there's been a lot of interest in P2x7 in regulation of ROS, but less on P2x4. So that would be worth looking at.
And Will Brackenbury asks, is altered lysosomal sodium and/or calcium handling involved in the P2x4 dependent mechanism regulating invasion?
I'm not sure what you mean by the handling of calcium. I mean, I think that when you change P2X, so we'd expect there to be less calcium efflux from the lysosome. There's also changes in lysosomal pH when you alter P2X4 receptor expression, which may also be affecting sodium levels. So
yeah,
I think that's very interesting and quite likely.
Okay, and for now at least, one last question from Michael. Could inhibition of P2X4 also be interesting in the treatment of tuberculosis, as manipulation of the lysophagosome is one of the primary modes of immunoevasion?
Yeah. I mean, there's been a sort of interest in P2X7 in treatment of tuberculosis, but I think that one would expect that enhancing P2X4 receptor activity and promoting lysosome-phagosome fusion would promote killing of the phagocytosed pathogen there.
Okay, thanks again, Ruth. That was lovely.